Hello friends, today we will discuss about prokaryotic gene expression regulation. In prokaryotes, it is generally done by LAC Operon. So Operon is multiple gene under single regulator or promoter. Bacteria use glucose as a preferred fuel for energy source and will use lactose only if glucose is depleted in the medium. तो फ्रेंड्स इसमें ऐसा होता है, आप इसको नाम लैक मीन्स लैक्टोज, लैक्टोज ऑपेरॉन, ऑपेरॉन का यू मीन्स ऑपरेशन, मतलब फॉर रिमेंबर पर्पस. लैक्टोज ऑपरेशन मीन्स लैक ऑपरन व्हेन इट इज फंक्शनल लैक्टोज विल बी मेटाबोलाइज्ड. लैक्टोज ऑपरेशन लैक्टोज विल बी मेटाबोलाइज्ड. तो सिंपल है. बैक्टीरिया मेनली ग्लूकोज को यूज़ करता है, लेकिन जब उसका ग्लूकोज ख़त्म हो जाएगा तो वो लैक्टोज को यूज़ करेगा. तो सिंपल है, ग्लूकोज प्रेजेंट लैक ऑपेरन नॉट एक्सप्रेस्ड, ग्लूकोज एब्सेंट लैक्टोज डाइजेस्टिंग एंजाइम आर इज सिंथेसाइज और इट मीन्स लैक ऑपेरन इज functional. Now we will go see this photo. There is I gene, there is promoter region, operator region, and then there are three genes: LAC Z, LAC Y, LAC A. These are in sequence. You can't arbitrarily go. First of all, there will be LAC Z, LAC Y, and LAC A. Okay. So I gene will form the repressor protein. Okay, repressor protein. ठीक है. That repressor protein normally bind to the operator. So RNA polymerase from the promoter region cannot go further, and thus the three structural genes are not transcribed, and the LAC operon is shut off. To become it operational, this repressor protein has to be bind with the lactose. Lactose will bind to the repressor protein. Now operator is free. RNA polymerase can freely go and transcribe these three genes and produce lactose digesting enzymes. Okay. And there is two important questions that is generally asked. Negative regulator of LAC operon R protein. Simply I have told you, because it will bind the operator and this LAC operon not work, so it is negative regulator. Then is positive regulator of LAC operon. It is CAMP, cyclic AMP. This you will understand in further. I will study. Now I think this should be clear. Now three situations can be there. We will discuss. First is glucose present, lactose absent. This is normal situation. Now in this situation, I have told you, bacteria will use glucose and there is no need of lactose digesting enzyme. ठीक है. तो now what will happen here? Now we will see. जो repressor protein will be there, that will bind to the operator. So there is no movement of RNA polymerase to ZYA genes and LAC operon is not expressed. So simple case. Now second situation can occur. Glucose absent, lactose present. Yes, that is the situation. It will be the functional, because अब बैक्टीरिया के पास ग्लूकोज है नहीं, तो वो अगर एनर्जी नहीं मिली तो ही कैन बैक्टीरिया कैन डाई. सो ही विल यूज लैक्टोज दैट नीड एंजाइम्स. नाउ दिस विल बी फंक्शनल. नाउ सी हाउ इट इज हैपन. तो लैक्टोज इज ए इंड्यूसर. Lactose will go and bind to the repressor protein that is formed by the I gene. Now operator is free. Now promoter can send its RNA polymerase freely to the three structural genes. Okay, and that can transcribe. But one important thing to remember, I have told you, I will tell about cyclic MP. Now second situation is, okay, lactose is present, glucose must be absent. When glucose is absent, there will be starvation like situation in the bacteria. Now in starvation there is a increased cyclic AMP, and cyclic AMP will bind to the cap protein. So now cyclic AMP cap complex is important for RNA polymerase to transcribe. Means without this complex, they along with RNA polymerase they also need cyclic AMP cap complex. Only then the digesting enzyme will be formed. So these two situations are important. Now you can understand why positive regulator of the LAC operon is cyclic AMP. This is because if only cyclic AMP is present, the in presence of RNA polymerase, the transcription will happen. Now third situation, that is very interesting situations. Glucose present, lactose present. Now bacteria has glucose bacteria has lactose. Now bacteria will use glucose. I have told glucose is the preferred source of energy. But as the as I have told, now bacteria will use glucose. Now, but lactose will bind to R protein. Operator is free. Yes, that is true. Now it means RNA polymerase go freely, but digesting enzyme not transcribed because there is no cyclic AMP. That is the important role of cyclic AMP. So this is about the LAC Operon. If you have any doubts, any comments, please comment below. I will try to give answer to every your question.